In this tutorial, we will discuss the clustering and marker gene identification module. The objective of this module is to use an unsupervised algorithm, that is, without a priori knowledge, to group the cells by cluster and to determine for each of the clusters which genes are markers of this cluster, that is, more expressed or less expressed in a cluster than in the rest of the cells. Beforehand, the project was loaded and the modules of HTO demultiplexing, quality control, gene selection, PCA and t-SNE or UMAP analysis were applied. In the gene selection module, we performed two analyses, one with the VST method selecting 1000 genes, and another using the mean var plot method. Each of these lists of selected genes was used in a different PCA. The first step in this module is to calculate clusters of cells. To do this, we first need to define the number of principal components we are going to use. If you have followed the module about t-SNE and UMAP, it is the same thing. We need to determine the number of principal components that contain enough information to be representative. The scree plot on the left helps us to see how much information is carried by each principal component, and to determine the value for which we consider that enough information is provided. For example, we may decide that the number of principal components here that are useful can be increased to 20. We enter the number 20 in the corresponding field. We then must choose the method used for clustering. There are several methods available. Here we will use the Leiden method. Next, we need to determine a resolution value. This resolution value is the sensitivity of the clustering. The lower the resolution value, the fewer the number of clusters, the higher the resolution value, the more clusters. We will first set the resolution to 0.2, then we will run the clustering. When the result is available, we can see that the cells are grouped by color, each corresponding to a cluster. There are currently 9 clusters in this result. The visual representation here is a UMAP, the one we created beforehand. It is possible to represent these clusters in a set of spaces that have been previously calculated. For example, in the PCA calculated with the VST 1000 gene selections, or in the PCA mean var plot, or in the t-SNE that was calculated from the VST gene list. If we choose the t-SNE, the map changes and the clustering adapts to the representation. It should be noted here that clustering and the dimensionality reduction t-SNE or UMAP are two different things. Clustering is not calculated from the dimensionality reduction. The two elements, clustering and dimensionality reduction, are both calculated from the PCA space. But they do not have a direct relationship with each other. It is thus possible to represent clustering in various representations, such as directly in the PCA space, by choosing from the menus the principal components of the X and Y axes that you want to display. Or, as we have done previously, to represent in a space of reduced dimensionality such as UMAP. Once the clustering is done, we can determine the marker genes of each cluster, that is, the genes that are more expressed in a cluster or less expressed in a cluster than in the rest of the cells. To do this, we must choose several parameters. First, we have to set the minimum fraction of cells expressing the gene. Here we have, for example, a value of 0.25. With this value, the analysis will only consider genes that are expressed in at least 25% of the cells in the cluster. All genes that are expressed in less than 25% of the cells will be ignored. Next, we need to determine the minimum log-fold change. The log-fold change represents the log ratio of the average expression in the cluster to the average expression outside the cluster. The analysis will only report genes whose log-fold change is greater than the value indicated. Here we will consider those with a minimum log-fold change of 0.5. We must then choose the method used to calculate these marker genes. There are several methods available. By default, the Wilcox method, that is, the Wilcoxon test, is applied. But you can choose another method. The help here gives you information for each of these methods. The last thing we must choose is whether we want to display the positive markers, or all the markers. The positive markers are the genes that are overexpressed in the cluster compared to the other cells. Negative markers are genes that are underexpressed in the cluster compared to other cells. Often, it is chosen to represent only the positive markers, that is, the genes that are overexpressed in the cluster compared to the other cells. This is what we will choose here. We can then launch the analysis. Once the result is available, we have several pieces of information at our disposal. In the first tab, we see the list of marker genes. This table only contains the first 200 most significant genes, but the whole result can be downloaded through the download button. In this table we see the name of the gene, the fitted p-value of the test, the average log-fold change, the percentage of gene expression in the cluster, the percentage of gene expression outside the cluster, the difference between these two percentages, and the number of the cluster in which this gene was found as a marker. In the second tab, for each cluster we will find the expression of the 12 most significant marker genes represented in a UMAP. The color is the expression level of the gene, which allows to identify if this gene is strongly expressed in the chosen cluster compared to the rest of the dataset. The cluster can be selected by its number in the drop-down menu. The third tab contains for each cluster the complete table of genes identified for that cluster, and the cluster, in the same way as before, can be chosen by the menu. Once the clustering is done and the marker genes identified, we can move on to the next module. Here you have two choices: either go to the differential expression module to push the differential expression analyses between groups of cells, or go directly to the interactive exploration, which allows you to explore the data in more depth. Each of these two modules is the subject of a separate tutorial video.